Yes. Okay, I'll go ahead and get started here on introductions. Although maybe I should wait till the speaker is back. I'll make it long. So it's my pleasure today to introduce Dr. Chip Petricoin. And he is at George Mason University right now, where he's a professor and the director of the Center for Applied Proteomics and Molecular Medicine. He obtained his doctorate at the in microbiology, actually, in 1990 at the University of Maryland at College Park. And then he went on to work for the FDA and the NCI, the National Research Council, as a postdoctoral fellow, and then as co-director of the FDA NCI Tissue Proteomics Initiative. Then in 2006, he went to the George Mason University as a chair of the Department of Molecular and Microbiology. Is that right? And he has numerous awards. Notably, I'll read you a few of them. The ASCO Keynote Lecturer Award in 2014, Harvard University and Children's Hospital Leading Edge Award in 2004, American Society of Cytopathology Basic Research Award in 2003, and the NIH Directors Award in 2002, and quite a few other awards for his proteomics and innovative applications and development of proteomics. He also worked with Lance Leota and Dennis Segroy when they were developing laser capture microdissection. And Chip is best known for doing the proteomics, the RPPA, which I'll talk some about today, on laser captured material so that you really have purified cells that you're looking at, not a gamush of different tissue and different cells in a tissue. He has over 375 publications, and he's on numerous grant review study sections for the NIH and the DoD. He's also on the advisory board for lots of different companies. I won't go through them all. And notably, he has 14 issued patents, not just patents pending. A bunch more pending, I should say. So he's been a collaborator of numerous people here at our institution, including Dr. Elias and I, for some specimens for clinical trial samples, but also for basic research samples in our breast cancer work. And he's also collaborated with Dan Theodorescu and back when Andrew Kraft as well, who used to be here, some of you might remember. And so it's a pleasure to have him here today to tell us more about his work. And the title is Protein Pathway Activation Mapping for Companion Diagnostic Marker Development Beyond a Genomic-Centered View of Precision Medicine Space. I think it hasn't changed. You'll see. Thanks.
Thank you very much. So hopefully this is on. Thank you very much, Jennifer, for that great introduction. And very happy to be here today to tell you a little bit about the work we've been doing in our lab to translate some of our technologies, our protein-based technologies, from the bench to the bedside. And in the course of the seminar, I'll hopefully give you some vignettes on how we're actually doing this. And then the last part of my talk, really kind of extending this to where we've been able to graduate some of these technologies to a real CLIA setting where we're able to use some of these approaches for treatment decision-making in a tumor board-based approach. So hopefully kind of give you the full scope of how we're translating this. So just from a disclosure standpoint, Mason, my university is very entrepreneurial, so they've been allowing me to do a lot of entrepreneurial activities, spin out companies, and also participate in advisory boards. So that's been a lot of fun and very interesting. Wish I would have taken a little bit more business classes in college, but I didn't take any. So I'm learning by osmosis. So when I was at the FDA, what kind of was front and center, especially in the Center for Biologics, which is where I was at the FDA, I was recognizing more and more that proteomics would be a necessary component of how not only the agency helped regulate drugs and biologics and devices, but also is becoming an important part of how we were understanding how the drugs worked, and also which biomarkers would be useful to understand how drugs worked. And if you look at, the compendium of FDA-approved drugs for cancer, by and large, most of them their mechanism of action is working on a protein target or protein targets. And of course, many of the biomarkers that we use to monitor disease, especially in a clinical situation, most of these biomarkers today are protein-based markers. Of course, this landscape is changing, but if you look kind of at the landscape right now, it is a proteomic landscape, if you will, in the context of what agencies like the FDA see. And of course, from a therapeutic standpoint, many therapies are protein-based therapies on top of it. So they can be both the drugs, the biomarkers, and even how the drugs work. So it's the working machinery and sometimes the therapy and very often the drug target itself. If you look at some of the initial, this is just some screenshots of some of the initial work that came out of the TCGA project years ago from Ken Kinsler and Bert Vogelstein's lab, where they were starting to do kind of the comprehensive sequencing of tumors. One of the big aha moments, right, that the field went through was at a molecular level. As you whole genome sequence these tumors, every tumor seemed to be different at a molecular level. Unbelievable heterogeneity in the molecular architecture of these tumors, which, of course, caused a lot of anxiety in terms of how do we effectively drug such a heterogeneous gemisch. This is not, every cancer was not a CML with a driving singular pathway. And this is still, a barrier to the full-scale implementation of precision medicine is the infrequency by which a lot of these alterations occur. And cancer ultimately is divided up into 2% of this and 3% of that. However, when they were going backwards and doing more of the computational analysis, they started to realize that while some cancers may have a PIK3CA mutation, and some cancers may have an AKT1/2 mutation, and of course the full landscape of every other types. Sometimes you're able to see that these things fell into pathways, and that there were pathways that may be shared. And these pathways, again, instead of targeting, a single gene, maybe we can target pathways that are shared. And this is becoming more and more, of course, a realized vision as we start to come up with both umbrella and basket trial designs where we're hopefully combining therapies and looking across tumor types. Well, these pathways, though, from not just a philosophical level, but from a practical level, it's proteins that form pathways, and metabolites that form pathways. It's the working products of the cell that actually work on each other. And so what we wanted to really think about is can we think about developing a proteomic approach that really understands the molecular architecture at the pathway level. And as I said, if we look at, even the package inserts themselves for some of these drugs, and I'm not going to pull up every package insert for every FDA-approved targeted therapy in the past 5 years, but if you go in, a lot of people throw their package inserts out. And I can tell you from an FDA perspective, we spent a lot of time on those package inserts. The package inserts are actually an approved entity as well as the drug. It's a cleared entity. If you go in section 12 of the package insert, usually clinical pharmacology, it tells you everything that the FDA and the drug company thinks how that drug works. And if you look at the MOA, the mechanism of action that's listed on these targeted agents, you'll see most often, these kind of explanations. And I pull up Afinitor just as an example, everolimus, talking about it's an inhibitor of mTOR. It's dysregulated in cancers. It binds these proteins, inhibiting this reduced activity of S6K and E1F or 4BP1 and EF4E, things that are downstream. You don't see a lot of discussion on this about genomics. There's not a lot of discussion about gene mutations and gene amplifications in these. And the reason for that is most of the time the MOA of the drug is against the protein and its protein activity. And this also gives clues from a pathway level of how these drugs may work. and then reverse engineering it perhaps gives clues on to what type of predictive markers could emerge in exploratory and qualifying analysis later. Now, of course, as all of you know that have pathways in cancer maps, the famous Weinberg map in every single lab across the United States, and this isn't that specific one, but you'll know, as we all do, that these pathways are not the simple cartoons that are drawn to display what we think how these pathways work. And of course, all of us are adding to the collective knowledge about these pathways as we go forward in time. However, being able to actually quantitatively measure the components of these pathways as growing in content as they are is now becoming a race. In some ways very akin to developing targeted exome panels looking at hundreds and hundreds of specific genes. We now, in the proteomics field, the race is on to develop platforms that can look quantitatively across hundreds and hundreds of very targeted specific panels of proteins and their modifications like phosphorylation, which are becoming exceedingly important, of course, because we have so many drugs that are kinase inhibitors and enzyme modulation inhibitors that are coming in, because while we're getting mutations in the underpinning architecture of cancer, the landscape of cancer is a genomic alteration-based approach. Really, at the end of the day, cancer is a proteomic disease. I'll say that because it's the functional architecture that provides a selection, the fitness test for the selective pressure that it's occurring is whether or not a cell has the ability to do something. So those genes are selected for, but the fitness test is through a proteomic lens, if you will. And so one way that this occurs, of course, are these modifications like phosphorylation that allow proteins to interact and talk to each other and pass information on. Ultimately, it's the field of signal transduction was driven by the initial phosphorylation analysis. And actually, the beginnings of that were in interferon alpha, where interferon alpha biology helped unwind what became the JAK-STAT pathway by George Stark and Jim Darnell, which started to reveal how these molecules in the extracellular environment were signaling into the nucleus. And largely driven by these SH2, SH3 interactions and the phosphorylation events that occurred when you had kinases acting on their substrates. So this is, in some ways, what we're trying to look at are these protein modification-based outputs and looking at kind of a pathway-based approach at doing this. Now, if you looked in the literature, there was clues to how protein modifications like phosphorylation may be adding value over and above what people were looking at specifically from a mutation-based approach. And so these are just a selection of some papers where these folks looked at specific cancer population. This is squamous cell lung cancer, where they looked at role for things like EGFR, KRAS, and PIK3CA, and predicting TKI, EGFR TKI efficacy, and didn't find any in these specific types of cancers. But when they looked, this is Dan Hayes' work, when they looked at phosphorylation, they found something very different. And so in this paper, again, they were looking at response to EGFR therapy, regardless of EGFR mutation status, and found that they were able, this is time to progression, looking at things like activation of EGFR and downstream activation like AKT as a signal integrator. Likewise, when you had the ability to go back retrospectively and look at and prospectively look at tumor samples that from patients that were given targeted therapy that today would never have received that targeted therapy. Like you very rarely now would have an EGFR wild-type lung cancer patient receiving things like erlotinib. That was not the case, obviously, early on when they were doing the initial signal finding before they realized that EGFR mutation was such a fantastic predictor in adenocarcinoma non-small cell lung cancer for this TKI. They would go back and look, and one of the things they found is that when you looked at phosphorylated EGFR expression in these patients that were EGFR wild type, they actually have a very good response prediction. The Kaplan-Meier isn't exactly overlaying on top of, say, what would be an EGFR mutant population, but they were able basically to pull out the patients that were phosphopositive had a much better response than the patients that were wild type and phospho-negative. This started again early on giving clues that there was a ability to measure phosphorylation over and above a genomics-only approach was finding responding populations. And this is a central theme from some of these papers where they, again, even in breast cancer, and Anthony and I have talked about this, things like PIK3CA mutations and AKT1 mutations have not turned out to be great response predictors for things like AKT inhibitors in the clinic. And there's a lot of work that was done by Sabine Lynn's group in Stockholm where they were looking at predictors for tamoxifen response and endocrine resistance, some very large trials where they were looking at genomic and phosphoproteomic analysis of these tumors. And what they found and kind of looked at in these papers, I thought kind of provocatively, for the identification of companion diagnostics, the focus should switch the analysis of activated proteins downstream in the AKT-mTOR pathway, which are associated with adjuvant tamoxifen resistance. And this was a series of papers that they published where they actually looked at phosphorylated components of the mTOR pathway that were extremely good at predicting patients' response to tamoxifen in over 1,000 patients. And these are some of the Kaplan-Meier curves where they were able to show that patients with low or high AKT-mTOR activation by looking at phosphorylation of the components had much different response to tamoxifen, essentially non-responsive here versus a difference in the Kaplan-Meier curves in the patients with low AKT-mTOR activation. So, there's, I guess, and I could go on and on as almost like teaching a class on all the background work on clues where people have gone back and looked one at a time at different components and seen evidence for addition of a proteomic view or a phosphoprotein view in cases where genomic analysis was done and you had or didn't have prediction emerge. So what I'd like to kind of postulate as motivation going forward for this talk is that under a microscope, the two cancer patients' tissues would look identical, perhaps sometimes histologically, but we know in a clinical trial may behave quite differently. So what we see, it doesn't explain, obviously, the full response. And even doing a genomic analysis may or may not explain the full response. We'd like to be able to effectively measure the components of this circuitry, if you will, this proteomic circuitry, and really look at this at a pathway level. So one of the barriers to entry in the field of proteomics is we do not have a PCR-like technology to be able to just chemically amplify low abundance signal. We don't have a ready-made tool chest of being able to go in and just amplifying low abundance proteins or low abundance phosphoproteins from clinical material, not a bunch of 225 flasks in the incubator filled with a couple billion cells, but basically having a few sections from a core needle biopsy, and that's all you have to work with for your part. How do you go into this and generate this type of pathway-centered view? So tissue input, tissue is the issue, as they say. So tissue input for a lot of these personalized therapy applications where the majority of the tissue is either FNA or core needle biopsy, especially in metastatic disease, is an input, is an issue. And you're going to have to reverse engineer your technology platforms to be able to use such small amounts of material. Tissue preservation is an issue, largely, especially in these large clinical trials where you have a lot of tertiary sites. It's limited to basically just plopping tissue into formalin. They don't have, carboys of liquid nitrogen or buckets of dry ice around, so that your biomarker at the end of the day has to be robust and resilient to the upfront processing. And of course, the tissue heterogeneity issue, both the clonal heterogeneity that everyone's kind of, focused on, and also just the heterogeneity of the tissue that you get in the amount of tumor cells or stromal cells or infiltrating immune cells or all the other components that make up tissue. And then from a precision medicine standpoint, the intoxication is looking at multiple markers, looking at pathways, looking at quantitatively. So translational-oriented oncologists that are integrating this information into, say, tumor boards, or they're trying to think about this from a basket trial standpoint, is you want multiplexed information, quantitative, and you need it being delivered in somewhat real time. So it can't be, we can do this, but it takes 3 months to do it. It has to be done, especially in a situation where you're looking at a translational output, say in a tumor board setting, needs to be done quickly. And of course, for proteomics and things like transcriptomics, we don't have a constant, stable archive like our genomic archive, which is obviously in flux from a mutation standpoint, alteration standpoint, but doesn't change as a consequence of how the tissue is fixed. That's not the case for proteomics and transcriptomics, because many times, you get the tissue out of the body, the tissue is alive on the pathologist bench. You have ischemia, warm and cold ischemia times as you collect the tissue, and depending on the robustness of your analyte, it can go up and down as the cells become hypoxic and acidotic. It's a wound at that point. And of course, you want to make sure that the molecular information that you're getting from that isn't a consequence of how the tissue is handled, especially if you're thinking about using this information in a CLIA setting where you're using it for treatment decisions. You don't want to have the information be changing just because of how the cells were treated. So there's an unmet need of fixation chemistries to actually stabilize ex vivo reactive biomarkers. And so we have done a lot of work in our lab, and we have a lot of funding from the NCI to try to develop reagents that can help obviate this fluctuating information archive. And we've done a lot of cold and warm ischemia analysis and published on this. We've gone to the operating room under IRB, approved protocols, and gone in and just sat there with a stopwatch and used all different types of fixation chemistries and different tissue types and looked at how does the signaling architecture and transcriptome change, and we have a window to work in that we can implement from an SOP standpoint. And generally, that window has fallen into about a 15 to 30 minute window that we have before things start to change as a consequence of the ischemia time. And that motivated us basically, and the NCI to fund us, to develop a tissue fixative that basically provides the yield equivalent to snap freezing the tissue, but actually has the diagnostic morphology and immunohistochemistry that's equivalent to paraffin, to FFPE. So basically, the NCI challenged us to develop a fixative that could give us FFPE-like histomorphology, but acted as if you were snap freezing the tissue from a molecular profiling standpoint and stabilizing that. Now, formalin penetrates tissue 1 millimeter an hour about. So as that tissue is plopped into formalin, don't think that those cells are just dead. So they're alive in the middle, and they're killed as a consequence of the diffusion rate. And every tissue type have different diffusion rates. More desmoplastic, the lower diffusion rates. So just plopping a piece of tissue, and sometimes even days later, that tissue in the middle is still alive, not happy. And I can tell you the transcriptome and proteome is completely different based upon that. Nothing to do with the biology of where it came from. You have to be very careful about this. So we try to develop chemistries that can help obviate this. And this is what we developed under NCI funding. We actually developed a universal fixative, and we assessed this for both phosphoprotein and protein on some very important proteins like HER2 and ER and PR for breast cancer, and other downstream phosphoproteins, and had that assessed by about 200 pathologists around the world. They did blinded calls by standard histopathologic kind of calls, and this had to pass muster on the immunohistochemistry side as well for both total and phospho. And finally, the NCI blessed it, and now it's commercially available. One of the interesting things about this fixative is that it's fantastic for studying bone disease because it does not require decalcification. And because it does not require decalcification, you can now do full molecular profiling of bone tissue with it, both RNA, DNA, and protein, which has allowed new use in clinical trials. So this fixative was developed under NCI funding and the IMAT program and is now commercially available, and we use it a lot for our molecular profiling precision medicine efforts. So, developing technologies to help preserve things like the phosphoprotein architecture is important. It has nothing to do with the downstream technologies that are used to measure it, but you're trying to mow down different components that are barriers to entry. And as I said, one of the biggest practical issues you have is that, you have a core needle biopsy or an FNA of the tissue itself, and of course, you're feeding at the trough with all the other omic technologies that are being used concomitantly that are sipping at the same tissue source. So it's not only a little bit of tissue, but it's a little bit of a little bit of tissue. And of course, you can't have your IRB-approved protocols be engineered to your technology. You have to engineer your technology to the little bit of tissue you have. So you can't say, I just require 15 core needle cores, and that's what I need. And that's what you have to do to the patient. It's like, no, you're going to get a couple cores, that's it. You're going to get a piece of it, that's all you get. And your technology's got to fit in or you're out. And that's the brutal reality in the precision medicine space right now. And you may be last in line, by the way. You have to wait until everything else is done. And so you have to develop technologies that can kind of meet that. And one of the technologies that's showing some pretty good utility is a technology that our lab invented a while ago back at the NIH called the reverse phase protein array technology, specifically to try to meet the demand of being able to do multiplex quantitative testing from very small amounts of clinical material. In fact, the originating paper that we used to describe the technology in Oncogene in 2001 is where we used this to kind of look at the invading front of prostate cancer cells to see if that invading front had a different signaling architecture than the trailing edge. So we had actually laser capture microdissect that invading front, very small amounts of tissue, and we had to use a technology like the reverse phase array to start multiplex testing the signaling architecture of that invading front. And this described the technology. Now, the array is really nothing more than kind of a nano dot plot, and it uses the same workflow as you would for immunohistochemistry. So it's a single antibody-dependent reaction. It's not a sandwich assay, and that's important because it actually gives you the ability, with validated antibodies, to do a lot of multiplexing. Because one of the biggest barriers to sandwich assays, you have to develop two very good antibodies that don't cross-compete with each other for the same epitope. So each analyte requires two good antibodies to work, and that is really limiting the field right now. You get a lot of basically epitope masking and competition that drives down the KAs, and that has been a big barrier to a lot of the antibody arrays. So this approach basically is where the material is immobilized on the array, and you come in with a primary antibody. That primary antibody is amplified by a biotinyltyramide reaction, like you would on a normal immunohistochemical output, like with a Hercept test. So you can actually use components of CLIA, CAP, and even FDA-approved assays like the Hercept test. We actually break apart the Hercept test kit and use majority of those reagent components for the reverse phase array. And that's important because that allowed us to graduate this into a CLIA environment very quickly. So over the past decade, we've taken this technology and kind of moved it through the usual chain of proof of principle applications, and then gone to preclinical translational applications, and finally deployment into clinical trials. Through that process, we've trained lots of labs around the country. We trained Gordon Mills' lab, for example, at MD Anderson, and he's developed a reverse phase array a lab down there. We trained his lab and his scientists there. We've trained a lot of them. So we're trying to get this technology out for use. One of the things that we're very happy about with this technology is right now it's probably the best, most sensitive technology for multiplex quantitative measurement of things like low abundance phosphoproteins in biopsy samples. Like I said, it requires only one antibody. It's able to look at very low abundance analytes, well below the detection limits of mass spectrometry, which my lab uses a lot of, or proteomics labs. So this is where antibody-based approaches have a lot better sensitivity than mass spec approaches for a number of these analytes. So you have to know which platform to use when you're trying to do it. So it has the quantitation sensitive and multiplex capacity that vastly exceeds Western blotting and immunohistochemistry. We've graduated to a CLIA platform. We've developed a whole series of calibrators and controls, just like you would have to do under a laboratory-developed test. And those calibrators and controls then are run every day as a proficiency testing, and so they're run constantly in our CLIA lab. And we generate then calibration curves. We know the precision and accuracy of every antibody, and then we're using this, as I'll tell you about at the end, for real-time analysis. So just like any other single antibody test, one of the biggest problems with that is that you better damn well have specific antibodies for your analyte, just like in no different than immunohistochemistry in that regard. Although immunohistochemistry has to its advantage localization checks so that you can kind of look to make sure that your subcellular or cellular compartment localization is what you think it should be. Here it's just a glowing dot on an array. So we don't have that signal check. But philosophically and really kind of logistically, operationally, it's like an immunohistochemistry workflow. As soon as the arraying is done, you have to have good performing antibodies. This is a huge issue, as everybody here likely knows. The antibodies that you buy commercially don't look like sometimes the Western blots that the company provides on their sheet. Those look really good, and usually you're holding your Western blot up against it, and it's like, I don't see the same thing. It's the same lysate. What's different? So we go through our own internal validation, and we require very specific, almost tiers of evidence of validating the antibody, single band, ligand-inducible, peptide-compatible, and also internally credentialed to making sure that you see pathway activation. So is that protein activation going up coincident with things upstream and downstream from it? So ultimately, when you go through this process, and we have gone through it, what it allows us to do is effectively go into these clinical studies and offer the ability to quantitatively measure hundreds and hundreds of proteins and phosphoproteins at once from very small amounts of input material. So it's very amenable to clinical trial type approaches, which is why we're using this in trials like I-SPY 2, which I'll tell you a little bit about. And I'm on two of the Stand Up to Cancer dream teams, the melanoma and panc dream team. So we're using that in the context of the clinical trial material that's coming out of those. You have to be able to use very small amounts in those trials. And as Jennifer alluded to, the technology certainly is being used for non-clinical material applications, and it's been very good at going in, especially in a systems biology type analyses where you're doing, transcriptomic and drug response data and metabolomic and multi-omic analysis of cell lines and manipulable systems. We published a paper looking at the NCI60 cell line, looking at interaction analysis of the reverse phase array with other types of omic analyses, and developed a series of therapeutic strategies from this that a number of pharma are carrying through. Likewise, many labs that we trained are kind of doing the same thing. Gavin Macbeth's lab had a really nice series of papers where they actually showed this in some of those manipulable systems. So, and let me also give note to the fact that these types of analyses are going on with a variety of laboratories right now. And Gordon Mills just had a very, very nice paper in Cancer Cell about 2 weeks ago, where they used this technology along with other transcriptomic, metabolomic, drug response data in about an 800 or 900 cell line panel. And that, if you want to see a tour de force paper, that's a very nice paper from about two weeks ago in Cancer Cell. So I'm just showing these as a series where RPPA, this technology has been used by a large number of groups looking at all sorts of correlative analysis in tissue and cell lines. And it is a tool that is now kind of part of the repertoire of our molecular profiling. Now, obviously, cancer is a product of the tissue microenvironment. This is a whole mount, prostate specimen that the motivation of me showing you this is that in this whole mount prostate specimen is a whole variety of cell types that are in that. And all of them can be found at once. And as anyone who's looked under a microscope of a tumor biopsy, you'll see every type of cell largely in that biopsy specimen that you can imagine. And I say that because we need to be able to understand the proteome in the context of the tissue microenvironment. And that brings up another important point for proteomics, differing than genomics, is that every cell type has its own unique proteome. There's no single proteome. The proteome is a fluctuating information archive that's cell type dependent, time dependent, and dependent upon the metabolome and the microenvironment. So it's a readout of what's happening in the cell at that point in time. So if you took this tissue and you ground up, kind of a grind and find analysis of the proteome or phosphoproteome, you will get a hodgepodge, basically, of all of these different cell types at once. And every one of these cell types have different amounts of the proteins you're looking at. So if you were looking at phosphorylated AKT, and you took this tissue and you ground it up, you would read out by the reverse phase array or many assays how much phosphorylated AKT was in that lysate, but it wouldn't represent at all the amount of AKT in any of these individual cells. You would not be able to accurately know, and there's no way to normalize to know that ahead of time. So it requires you to go up front and really kind of collect the target cells to know what's happening in those cells. Very different than when you're looking at, say, a rare mutation, and it's more of an analytical consideration of what your sequence coverage is and your repetition to be able to know from an assay validation standpoint, I can see mutated EGFR when it only 1% of the cells, because it only occurs in a single cell type. The proteome is very different, because every cell largely has some concentration of every protein in it. There's really hardly any example where you only have one cell type, only have 100% of a given protein. If it happens, it's very rare. So technologies like laser capture microdissection become an important part of the workflow of providing a proteomic readout for precision medicine, because we have to overcome not just the clonal heterogeneity issues that occur, but also just the heterogeneity issues that occur in a tissue specimen itself. So you want to be able to go in and pick out these specific cells and use that input to kind of understand what's happening to the signaling architecture in those cells. Our lab at the NIH and Lance Leota's lab really was the inventing lab for laser capture microdissection technology, and this is certainly part of our workflow. And we had to publish a series of papers where we actually had to show that the laser itself did not destroy the phosphoproteins, didn't disrupt the phosphoproteins by going through the tissue procurement process. We went through a whole series where we just microdissected, if you will, all the tumor cells that were on the slide versus microdissecting single individual cells versus lysing kind of contiguous sections directly without laser, and show that there was no effect. We also published a series of studies as a result of us being told by different labs that some of the TCGA RPPA data was discordant from what they expected. And our lab didn't produce the RPPA data for TCGA, but we were highlighted to the fact that there was some discordance there. So we went back and actually looked at some of the samples that went into TCGA ourselves and performed an experiment where we actually showed that the fact that the tumor cells in TCGA were not laser dissected, that the RPPA data could be inaccurate sometimes, depending on the analyte that's there. So we took a large variety of tissue samples. These are GBMs, where we basically breadloafed the tissue. We took the tissue samples and we basically separated them out, microdissected versus non-microdissected, and arrayed them at the same time. If the information was the same, we would have seen it. And it was not. And then more definitively, we went to look at what was described to us as being discordant, which was P10 and EGFR phosphorylation that was looked at by these investigators. We found, and we reproduced what they found, that basically that only in the microdissected sample did you get the expected concordance between, say, P10 loss or mutation and P10 expression levels. both at the mutation status and the copy number status. So this was the matched slide lysates down here, undissected. So only when you microdissected did you get the expected differences. And in fact, if you looked at the TCGA data, they found the same thing, that we basically did not see the expected differences. And this is because, of course, is that P10 isn't a protein, like I said, expressed in lots of different types of cells. It's not owned by tumor epithelium. And so if you have a tissue sample that has P10 expression in other cell types and you ground it up, you could have loss in the tumor, but it would be swamped by the amount of P10 in other cell types. And you would have missed entirely that concordance. So our point is, well, only takes a couple things to miss, and then it calls into question every analysis because you don't know ahead of time what you've missed and not missed. So everything we do is through this laser dissection process. So I'm going to toggle over for the last kind of 20 minutes to describe how we're using this in these different clinical trials. So one clinical trial we're using this a lot in is the I-SPY trial series. Dr. Elias obviously is an investigator in this trial. It's a lot of great clinical investigators get to collaborate with. This, in I-SPY 1, is basically a neoadjuvant cancer trial where we're able to get biopsy specimens before, during, and after targeted treatment in a neoadjuvant setting. And this formed basically the motivation to go into I-SPY 2, was developing this kind of I-SPY clinical trial sandbox. And we started looking at the pretreatment biopsy sample just to develop the signaling architecture of breast cancer at the molecular level. And so this was some of example data we published in Cancer Research. We're just looking at the signaling architecture of laser microdissected I-SPY breast cancer samples. And what we found very quickly is that we could see clustering, red is high levels of activation, green are low levels. This is an unsupervised analysis. And when we looked at different phosphoproteins and proteins in specific pathways that we knew were drug targets for things that were going to be coming into I-SPY 2. So we looked at I-SPY 1, motivated by drugs that we knew were going to start coming into I-SPY 2. We looked at those phosphoproteins based upon those drugs that were coming in. So we knew we were going to have AKT, mTOR inhibitors, and we knew that we were going to have HER2 inhibitors coming into I-SPY 2, so we developed content around that. So you can certainly see patients that had, high levels and low levels of these pathways are activated at the pathway level, and I'll tell you what that means. And these were, this is an example where we just looked at the triple negative breast cancer subset of I-SPY 1. And when I talk about kind of a pathway level view, I basically mean that we're able to build content where we have some redundancy at the pathway level that give us the ability to measure whether or not the pathways are actually activated. So what do I mean by that? Well, I mean that even though this is a snapshot in time, and we're kind of getting a population averaged amount of cells in a biopsy sample, we expect if things like EGFR are activated or AKT are activated, we would see evidence of this downstream with the substrates that they act on. Even though this is a time snapshot, no synchronized cells, they're all at different parts of the cell cycle. In general, stochastically, you would expect if something's activated, the nearest neighbor hypothesis comes in, and you would get a stoichiometric high increase in the next nearest neighbor. And we see that. So you see things like EGFR activation coincident with SHIC, IRS, and adapter proteins. This is unsupervised, so we didn't train it to basically cluster together. We see things continuously, things like here's AKT and its direct kinase substrates clustering together because, of course, if their patients are high or low in that, they basically drive the coalescing of these events. We published about 12 papers on this, and it's a very good way and a consistent way of actually doing due diligence of the final data. We were also able to do diligence on our data by comparing the reverse phase array data to like FDA gold standard ways of measuring an analyte, like HER2, FISH, and IHC testing. So in I-SPY, we were able to credential the RPPA data against this. So here's a rank order plot of the HER2 quantitative data on the RPPA, and then the calls made by central IHC. So you can see in green are the HER2 positive IHC patients, and in red are the HER2 negatives. And this explosive dynamic range of the HER2 positive population, these are all 3 pluses, is important because we're going to show data later on where this is clinically relevant, is an example that we see a lot. So I could show you almost the same plot with PD-L1 in lung cancer. It looks almost exactly the same, where the dynamic range and what is called positive is massive compared to the small dynamic range in the 0 to 1s. Similarly, here's kind of concordance with fish. And again, similarly, you have the ability of the patients that are obviously positive are all high, and the patients that are low, except these 3 patients here actually were just concordant. They were low by our assay, but fish positive. What's interesting is now when you look at this, so keep this pattern in mind. And now the next slide I'm going to show you is not measuring HER2 total, but actually measuring whether or not it's activated and phosphorylated. And I want you to keep your eye on this population of patients down here that are HER2 negative by FISH and IHC. They're HER2 low by RPPA. I'll tell you they're also HER2 low by mRNA expression, and they're HER2 wild type. So they're not mutated, amplified, but some are activated. There's a portion of these, so now we're looking at phosphorylation of HER2. So the same patients that are all here that are FISH negative, there's a portion of them that are phosphorylated. And they're phosphorylated to the same level as the HER2 positive, frankly, activated FISH positive, HER2 positive population. These are these 3 patients that were also low by the total. They're also low by phospho. These patients right here are the ones we're really interested in, because I can tell you that none of these patients are getting HER2-directed therapy. They're HER2 negative, and they're not amplified. They're not getting in any trials, but yet they're activated to the same level as the HER2 positive group. All of these patients are getting targeted therapy, HER2 directed. None of these patients are, and yet there's a population of activation similar. And we went and did genomic analysis to see that there was no correlation. So when we published this, we published that not only were they activated, we saw this on a very large number of study sets across 3 different labs in Europe, and a study set in Europe was used as a confirmatory validation set. We also saw evidence that there was heterodimerization with EGFR, because not only are these patients high for phospho-HER2, they were also high for phospho-EGFR, and they had similar downstream signaling architecture. So they had high levels of SHIC, FAC, and STAT3. So this is the IHC positive group. This is this group that's basically IHC negative but phospho high, and this is the true IHC negative phospho-HER2 negatives. So these 2 are the ones that kind of looked alike. And the oddball out was the true negatives group. This will be important as we talk about neratinib response later. So about 10% of these patients were FISH IHC negative are actually phospho-HER2 positive and are pathway activated. Interestingly enough, Soon Paik published in the New England Journal in the B31 trial that about 10% of patients that were HER2 negative received benefit from trastuzumab. So one can speculate that maybe that these patients were maybe phosphorylated. So again, in the framework now of I-SPY2, where we're getting the same types of samples, but now the patients are being put on a new adjuvant therapy that includes experimental agents prior to anthracycline, we're able to compare the PCR rates of these to the pretreated biopsy specimen. And as you know, or likely know, that there's eligibility criteria that are based on mammoprint as well in I-SPY2. So these patients are coming in, and they're being given a variety of different types of drugs with different molecular mechanisms of action. And the first drugs to graduate on the I-SPY were neratinib, a pan-ERB inhibitor, basically an ERB2 EGFR inhibitor, and veliparib. We're working on these other ones now. Now, neratinib graduated, as you likely know, in I-SPY2 in the HER2 positive population based on an estimated PCR of 55% compared to 32% in the control arm, which were trastuzumab treated and I guess Perjeta treated at some point patients in the control arm. And the probability that neratinib was superior to that treatment was 94%, and this probability of success in phase 3 that Don Barry uses as a Bayesian output is what drove the graduation of this drug in the HR negative HER2 positive population. By the way, the most infrequent of the molecular subtypes. So I don't, it's not treating, it wasn't shown, there was no success shown in a majority of the breast cancers, I guess is another way to say it. So when we looked at what was predicting PCR in these patients, and we looked at a number of the EGFR HER2 signaling cascade as a qualifying biomarker, we actually found that we were able to predict PAPCR in patients, pre-treatment biopsy samples from patients with activated EGFR and HER2, and things like SHIC downstream. So this provided a motivation for us to go in and look at how much prediction that was giving. And so if you looked at these patients that were EGFR phospho high or low, we actually found we could add value to the graduating IHC marker based upon whether or not they were phospho high or low. So when activated EGFR were added to the graduating subset, the predicted probability of phase 3 success was 90%, which is comparable to the graduating arm, but we increased the prevalence of the biomarker positive patients by 50%. And I'll get into that a little bit more. Because when we started to look, if you look back here, one of the interesting things was that while this was patients that were graduating based upon this PAPCR status, we found not just phosphorylated EGFR HER2 being predicted, but also total levels of some of these proteins also being predicted, like total HER2. So well, that's interesting because these are already being selected in a HER2 positive or negative IHC setting. So when we looked at the quantitative aspect of HER2, remember I said that you have this huge dynamic range of HER2 expression by quantitative means that was all 3+. When we actually started to look at the HER2 positive patients in neratinib response, we saw kind of a stunning scatter plot here. So you found that almost all the PAPCRs were at the higher end of the quantitative HER2 assay. So by a cut point where you don't miss hardly any of your PAPCR yeses, do you basically get rid of a lot of your HER2 positive non-responders. So these are all patients that are HER2 positive by FISH and IHC, but yet the responders were all on the higher end. So a simple quantitative HER2 test here was actually able to add value potentially to a cut point that could be used. So not to belabor the point, but the main thing here is that we were able to find activation levels of these receptors that were based on the MOA of neratinib were indeed predictive and were able to add value to the exploratory analysis. Now, what was interesting about neratinib is it was also given to triple-negative breast cancer patients. It was given to every molecular subtype. And what emerged in our analysis of neratinib in the triple-negative population was really stunning. Now, we went into this population with a qualified hypothesis that measuring the activation state of the receptors, EGFR and HER2, would still be predictive of response based on what we found in I-SPY-1. We found this population of patients in I-SPY-1 and published on it that was negative but phospho high. So we felt like, aha, now we finally have a drug that was given to these patients, to a patient population, and we have response outcomes data on these patients. It's not just observational. And what did we find as the sole predictive markers for neratinib response was phosphorylation of HER2 and EGFR. And the triple-negative population was able to identify patients responded to neratinib compared to the control arm. And what's, again, very interesting, so this is a two-way scatter plot of activated EGFR and HER2, and this is the control arm, and this is the treated arm. And you can see all your PAPCR yes patients are in this upper quartile. They're basically double positive. So if you were high for EGFR and HER2, you essentially, almost all of them achieved PAPCR. So basically being able to develop a 2-point cut point was able to identify a responding population. And in fact, that responding population looks really good statistically. So in the bottom, the PAPCR rates in the kind of the IHC determined population. On top was our PPA population. So our PAPCR rate is 77% in this selected population compared to the 55% in the graduating arm. And that likewise a very similar PCR rate in the control group. But the prevalence is what makes it very interesting. So the biomarker positive population that's high in the triple negative breast cancers, that's phospho high, is about half. About 43% of the patients are activated. And we get about a 70% PCR rate in that. Well, 77% of triple-negative breast cancers is something to consider in prospective analysis now. And this is exactly what we're doing. Puma is running a prospective clinical trial with Joyce O'Shaughnessy, basically repeating the I-SPY 2 analysis. And they're doing it with a CLIA-based test that's being developed with it by Avant Diagnostics that would be potentially part of a package insert in a phase 3 setting later. So this is, we have a signal. No other markers have emerged. No genomic markers, no other markers have emerged predicting response to neratinib in the triple-negative population. Only phosphorylation. And again, it's something where now we're going to be prospectively validating this to see if it continues to hold water. We also found a similar instance with veliparib. And so what we found, again, is on the backbone of what was found by an IHC-based determination. Veliparib graduated in the triple-negative breast cancer patients with a 52% estimated PCR rate compared to 26% in the control arm. And if you look again at the quantitative, again, so I'm kind of giving you the Cliff Notes version of our analysis, one of the markers that emerged as a predictor was total HER2, which you would have been like, well, how is total HER2 emerging as a predictive marker in an ostensibly already HER2 negative population? These are triple negative patients. How can quantitative HER2 be useful? Well, here's where the quantitative aspects, again, reveal that a lot of the patients that are achieving PCR are at the low end of the HER2 level. If you have a lot of HER2 in a low amount, if you have a lot of a little, you are not achieving benefit. So we're developing cut points to be able to help us basically find that these, cyclin D1 was another one that emerged, but HER2 is one we're concentrating on because we can toggle it against an FDA-approved way of measuring it. Again, if you look at what was happening in triple negatives in the predicate based upon an ERB2 cut point by RPPA, we're finding patients and enriching for them compared to HER2 by itself. And point mutations are not part of this. So it's not like these are all HER2 mutated or activated like Matt Ellis has found. These low, too infrequent to be important. So I'm going to finish up at the very end here by talking about how we're taking this as a CLIA test to looking at how we're treating patients in the setting of metastatic disease. So this trial, called the Side Out trial, and we're hoping that Peter Cabos comes on to Side Out 3 here at University of Colorado, is where patients are coming in who have progressed off of standard of care therapy, are getting a multi-omic profiling done, and that information is going to a molecular tumor board, and that tumor board then decides what therapies to give, and then we follow patients. So a very standard kind of basket trial-like design, except it's only using FDA-approved inhibitors that are off-label. So no experimental therapies. And we're using, as a predicate, the Dan Van Hoff's Bisgrove trial, where he was using this progression-free survival interval change to look at patient-based changes in PFS as a result of molecular profile induced benefit or not. So in the Side Out 1-2 trial, we had a number, about 7 different centers that were collecting patients on study, and we were profiling them. Caris's Molecular Profiling Institute was involved. We were doing the phosphoproteomic analysis, and there was exome and IHC, and our phosphoprotein was all being done. Again, we can do this because we were able to develop cut points. We had population data. We have calibrators, and we were able to do this under a CLIA setting. And what we're able to do is, just like any other assay in CLIA, we have a patient population that we run all the time. We are able to develop cut points, and we can compare an incoming patient to a population to know what is high or low. Just like you would say, how do you know if it's IHC positive or not compared to what? So it's the compared to what that you basically develop under a CLIA LDT. And then we can match that to drug targets and activity levels. And this is what we do in our study. So we go through a soup to nuts microdissection, reverse phase array analysis of a targeted subset of endpoints, which I'll show you. And we can turn this data around actually in about 72 hours. So from beginning to end. And we deliver a report back to the physician and the treatment selection committee that basically is in a series of phosphoproteins like EGFR and HER2 and HER3 and AKT and mTOR on specific drugs that have targeted these specific endpoints. We're also going to be layering on things like palbociclib, where we're going to be hopefully measuring things like CDK4/6 kinase substrates. For the purposes of the rigor of this study, we said that activation or not activation is that the patient had to be in the highest quartile of the population, of its predicate population. So meaning, if we're going to say it's activated or not, it had to be very high. And not only did the drug target have to be high, but we had to have evidence that at least one member of the pathway downstream had to be activated. So if we saw HER2 activated but no EGF, no AKT or ERK or mTOR activated, then we would not call that pathway activated. It had to be kind of a pathway activation. And so this is this difference in PFS interval by prior therapy to the molecular profile selected therapy, and it has to be a 30% increase to be considered success. And as we reported in ASCO, we actually achieved our primary endpoint. About 52% of the patients actually had an increased PFS of greater than 30%. We had some, I don't know if we would term them exceptional responders, but certainly very long-term responders, where we had this one patient who had their PFS interval was about a little less than a year, and then the one on therapy went out a little bit over 4 years on study. And so this motivated us basically to go into SIDE-OUT 3, and we found that we could, in fact, do a multiplex genomic proteomic analysis and turn this around in the aggregate, and the time period that the feasibility of such approach would be achieved in a real-world setting. It also allowed us to go back and do some very interesting analysis. We were able to get whole genome and phosphoproteome in the same patient and compare things that occurred at the genomic level compared to what we found at the phosphoprotein level. And those have been very interesting for us. We're going to be writing a paper up on the full case studies of these hopefully in the next month. So the SIDE-OUT 3 trial is starting, and this is where we're actually layering on this multi-omic approach on top of trying to find a palbociclib response predictor. Again, one of the interesting components of the SIDE-OUT 3 trial, because of our fixative, will be to include patients who have bone-only disease or where the bone metastases can be obtained. And that'll be the input for the molecular profiling. I do want to finish with some analysis we've done where we're actually able to use proteomics for something only proteomics really can be used for. And that is measuring whether or not you can measure real-time pharmacokinetic changes in the drug target. So you can look at what did the drug hit the target. So this is a study, I should have not toggled past this, we used basically imatinib and panitumumab in colorectal cancer patients in the liver metastasis. We're actually giving Gleevec and EGFR inhibitor. We're going in over and over again in the same biopsy and looking as they got different therapies. So they got on study, they got a dose escalation of imatinib, we got the pretreatment biopsy, then after imatinib, and then when they got both. And that allowed us to kind of look at the drug hit the target. So these are like pre and post paired samples. We could see the post-therapy biopsies. You could see downregulation of things like PDGFR and KIT and ABL, and the downstream things like AKT and mTOR. So we're able to actually use this to see the drug hit the target, kind of kinetic analysis and pre-post paired biopsy samples. And anecdotally, in these patients that achieved the best rate of change, actually were the few that actually had a response. Did not hit resist criteria were not met. We saw these anecdotal examples where in patients that did have really good response, we actually saw initially then this targeted change. We're also able at the same time to look at these sequential biopsies and see evidence for pathway modulation and maybe things that are adaptive or changing as a result of the drug. And we have to see whether or not resistance is part of it. We're also able to see whether or not the molecular architecture was different. So that from a tumor heterogeneity issue, we're able to go in and see if you sampled over and over again from the same lesion, did you get different results? And largely, the patients all clustered together. So this is just the same patient where we sampled at day 1, day 30, day 1, day 30, day 1. This one we actually had end of treatment. And they were all clustering together, and we looked at, in this paper, more kinetic analysis of the drug targets, and look really good. So the future of companion diagnostics, as I see it, where proteomics brings into the picture, is the ability to measure so many different drug targets at once from very small amounts of material that are all based upon the MOA of the drug. We're going to have lots of FDA-cleared inhibitors coming in. You're going to have a whole selection to choose from, and there's going to be a need for multiplexed analysis, even on-label drugs. I mean, you look at things like kidney cancer, there's like 7 or 8 target inhibitors are FDA approved. At some point, we're going to need to measure these different drug targets ahead of time to know which FDA-approved drugs to use. So this is where some of these technologies like MS or layered IHC or nanocapillary, you're simply going to meet a physics problem of being able to effectively measure so many different things at once from so small amount of material. And to date, the RPPA technology is the sole technology which has allowed us to be able to look at so many things at once from such small amounts of material. Ultimately, I think that this is where we're trying to find a pathway-centered view of cancer. This isn't just my vision, but shared vision by lots of people. We're starting to maybe redescribe cancer, not by site of origin, but by pathway activation. And perhaps in the future, as part of a pathology report, we'll have techniques like the RPPA, part of the usual workup of the clinical pathological analysis of tissue, and maybe even providing the oncologist with individualized activation maps that can be used in the future by the oncologist to make treatment decisions. So I'd like to thank you guys for listening to me, and I'd like to acknowledge the many, many people in our center that do all the work and that I get the honor of talking about and representing their work. I want to call out specifically Julie Wolfcool and Valerie Calvert and Mary Elena Piraboon, basically who represented most of the 90% of the work that I talked about today, and then my colleague Lance Leota, and all of the different funding agencies that have provided the funding for me to be able to present this work to you today. Thank you very much.
One is how many cells you have to laser capture in order to get a good signal.
Yeah, so what we found is you will get, from a tumor heterogeneity standpoint, where lesional, you can go down to a point where you can grab very small areas, and they will be different area to area. That difference there is actually not that different if you start doing kind of proximity dendrograms compared to like other patients' tissue or even interlesional work. So that being said, what we try to do is microdissect about 15,000 cells worth, which represents enough different, spatially different cells that we get kind of a gestalt of the tumor epithelial environment in a pretty good proximity to each other. So we about 10 to 15,000 cells.
The second question is, if in I-SPY 1 or 2, when you look at the control arms, particularly, let's say, in the HER2-positive group, you get Herceptin. Do you see actually a difference? Because I would expect that the Neratinib, because it's an irreversible EGFR and HER2 inhibitor, that the downstream and the phosphorylation will be particularly important in mechanism of action, whereas with Herceptin, it may be total HER2 expression that may be much more important than in fact activation. Is this true?
I think so, but we haven't had the evidence yet. One of the things I've been wanting to do since I-SPY 2 opened was to actually look at response predictors for the control arm. Right. And as you know, one of the limitations of I-SPY 2 is the numbers, especially the control arm. So we've been really pinched by small number analysis. So I've actually had to wait for enough control arm patients to be accrued to actually look at the PCR rate for the control arm of the HER2 control arm, the HER2 positive control arm. And then I would really like to look at the pre-treatment and on biopsy sample so I could look at how the pathways, drug targets were affected to see if that modulation was affected by a real TKI compared to, say, an antibody like trastuzumab. Or other MOAs that, like you said, that the drugs themselves may have them. Yes, whether absolutely. So I don't have any data to speak to that at this point, unfortunately. Any other questions?
I was wondering, the triple negatives that were high in phospho, or EGFR, sorry.
Mm-hmm.
Did they fall into any particular subtype of triple negative, like the type 4 subtypes?
No, so just, yeah, it was done actually. We worked with Denise Ching and or Christina Yao and Denise Wolf that were doing a lot, and Laura Vanterveer, they're doing a lot of more of the genomic and genetic subtyping. And there hasn't been anything that has fallen out yet that has described that population, that phospho-EGFR, which is the same population as phospho-HER2, you're kind of describing the same. Hasn't been, there's no description of that kind of genomically yet. Not to say that there may not have something fall out, but at this point, nothing yet. It's fascinating. Thank you very much.